Hi, welcome to Brazil. My name is Georgia Sinimbu and I'm a grad student at the University of Utah. But I've been working in Brazil for my research. You guys are about to engage in my research as my field assistants for this expedition. I hope you're all excited. I'm very excited to have you here with me. So first of all, I'm going to tell you a little bit what is it need to arrive here, and then we're going to do a little trip. Actually, not a little trip. We're going to travel all the way to the forest, and once we're in the camp, I'll tell you about my research. Okay, right now, at this moment, we are at INPA, which is the National Institute for Amazonian Research, and it's at Manaus, which is the capital of the state called Amazonas in Brazil. So this is all the way north in Brazil, right in central Amazon. Since my undergrad, I've been working at INPA to do research in the Amazon, and I have to admit I felt in love with the forest, and I'm sure you guys will agree with me once you guys get there that it's just amazing. So to do research in Brazil, and I believe that in many other countries it wouldn't be that different, you need a lot of paperwork. So for instance, in here you need permits for collections, permits for transportation. You have to have a registration at the Brazilian Natural Resource Institute. Besides that, you have to be associated with a research group. So in my case, I'm associated with the DBFFB project, which stands for Dynamic of Biological Force Fragment. So it's one of the longest projects in the world that is trying to measure the effects of fragmentation in tropical forests. Because I'm Brazilian, it makes it a little bit easier for me to have those paperwork, but it's not that easy. So preparation takes, for a trip, preparation takes about 6 months to get all the paperwork, and I am guessing, because I'm not pretty sure, it would take something like a little bit more than 6 months to an American be able to come here, because on top of all the paperwork for the work itself, you need the visa, and it's not a tourist visa. You need a working visa. You watch out for that. So preparation takes a lot of time, and this is what I'm telling you just for paperwork, right? But you still have to prepare all the research you're gonna do, because it's such an expensive trip that you don't want to be losing time rethinking your stuff. So you need to come prepared. I had a professor that used to say that for each day in the field, you need 10 days in the lab of preparation. And I would agree there is something like that, although once you get in the field, it never works. There are a lot of other stuff that we just can't control. And then you need money for all that. Oh God, money, that can take a lot of time. And I'm telling you by personal experience. For instance, for my trips, I spend around $1,300 per month. So on that, I include food for every trip, and the transportation between the institute and the camp. I have to pay for field fees, and I also have to pay for the assistant. It's just a lot of money, and then you have a lot of things to put together, right? And then this is my, I don't know if you can see, so this is my gear, like gear stuff and food for the 10-day trip, and we're about to start, and I am waiting for my assistant to be here and to start packing everything in the truck, and we can leave.
Queima!
after 3 hours of road, we reached the entrance of the camp, but we still have 700 meters till we get to the camp. And now we have to carry all this stuff here all the way down. Welcome to Camp 41. We're finally at Camp 41, and this is the place we're going to spend the 10 days of this expedition. And this time, who's going to assist me in the field is Adriano.
Hello.
So Adriano just finished his master, and he worked with methods for sampling ants, and mostly leader ants. And I'm counting on him to help me identify some of my ants, right?
Right.
Okay, so let me show you the camp where we're going to spend our expedition. So here is the social area. It's the square. When there's a lot of people in this camp, that's where the social interactions happens. And there's parties here too, if you can't believe that. So there are 3 buildings here, and these are the rooms, let's call it rooms. And this is where we sleep in and spend part of the time here. So we usually sleep in hammocks, and it's quite comfortable once you learn how to sleep in a hammock. So the camps also have bathrooms. So here we separate toilets from the bathing area. So we have to take this trail to reach the bathroom. And these are the bathrooms, and right over there is the generator. It's the machine that will generate electricity for us. We usually use it for 3 to 4 hours per day, and we also use it to charge the water tank. Worse than not having electricity is not having water. It's very hard to deal with in a camp that you don't have easily access to water. So here are the proper bathrooms. This is where we are going to take our shower, but I rather have my showers on the swimming pool. And this is the swimming pool. It's quite dirty because I haven't been able to clean it very well. It's too big for a person alone to clean. So this is our water tank. It can hold 2,000 liters water, and it's enough for 2 people during 4 days. And this is the kitchen and dining area. So when it's just me and my team, I usually use these tables for my lab stuff. So this is where I work, and this is the kitchen. We have all the necessary tools to do good food. So we usually eat very fancy here whenever I'm in the mood to cook fancy, which is not always, because once we're back from the field, we have to cook, and then sometimes we're just so tired. These are the storage. We have to leave the foods hidden, otherwise rats and marsupials will take all our food. And those are the fridge. That's where we store the meat and some of the vegetables that need to be in the fridge. So this camp can hold up to 35 people comfortably, but I've been here with something like 40 people, and it's okay, it's not pleasant, but it's a quite safe place. So something I forgot to show you, once we have electricity, that box all the way there in the back is the radio that we use to get contact with Manaus. So whatever we need, if we have any emergency, we can just call them and get their help. But remember, I told you it's 4 hours far from Manaus, so we don't want to be in any kind of trouble. A lot of people come and go here. This year, I am the person that has spent the most days in this camp. So I think I am about to complete 80 days in the camp. Not straight. So usually what I do is 10 days in the camp, 4 days in town, back and forth. But since the beginning of my field season, I've been here for about 80 days. So I feel this camp is my house because I've spent more days in the camp than in the town in Manaus since I'm in Brazil. So I like to take good care of this place. So things that we need to do is just keep places clean, especially the sandy areas. So we have to sweep because it would be very hard to see a snake if during the night, and it's very common to see snakes here. And usually we have this one called jararaca that I can't remember the name in English, and it's a poison snake, so you don't want to be in danger with the poisonous snake in here, right? Remember, it's 4 hours far from town. And the other day we were just sitting here eating dinner, and then this snake just fell out of the roof because it was hunting the frogs. So the project to which I am associated here in Brazil have many camps. This is just one of them, but this is the most beautiful one, and I feel quite fortunate to be able to be here. And so it's 7:30 in the morning and we're ready to go to the field. So we have a 35 minutes walk till we reach the reserve where we're gonna work, but we have to walk through the forest and then a little bit through the road. So are you guys ready for that? Shall we? We just found army ants. Check this out. So they usually don't have a fixed nest. They are nomads. It says the English name. So they will move once in a while, and what they do, they search for their prey in huge numbers, and they can be quite scary for the other living beings. I've seen once a frog being eaten by those ants. So here we are in a permanent plot where we measure dynamics of the forest. It's a 25-hectare plot where all the individuals with 1 centimeter above diameter height, which is like, I'm too small, so it'll be something right here. And if it measures more than 1 centimeter, which is something a little bit thicker than my finger, then they are included in the census. So all the plant individuals are measured and they have a tag. For like this one here, it has this tag here, and I can just look up on the This plot is administered by the Smithsonian Institute and INPA. So basically the plot looks like this. It's many 20 by 20 meters subplots. So for instance, right now we are right here in this subplot. I don't know if you can look at it. So here is the plateau area, and here are the slopes. I do my work here just because I don't have to deal with plant identification, because in the Amazon we have lots of species. For instance, in each one of those 20 by 20 plots we have in average 300 individuals. Can you imagine 300 individuals? Imagine something like a small auditorium, maybe a little smaller than ASB 220, and fit in there 300 trees. I'm not talking about plants, I'm talking about trees. So imagine trying to fit 300 trees in ASB. It's a lot, right? The focus of my PhD is ant-plant association, but I'm more interested in what we call facultative associations. So which are associations that are mediated by sugar rewards. So for instance, let me show you. So this is Inga tibaldiana, and you will meet Lisi Colli probably, and this is her preferred Inga in Brazil. And these Ingas, they have this little nectaries here in between the leaflets. I hope you can see. So this one, this one, this one. So these are called extrafloral nectaries, and they are usually active during leaf expansion, when leaf is more vulnerable for herbivores. Let me show you some. So this is another plant, and these are the mature leaves that already attained their full size, and these other ones here are the young leaves. They're the leaves in expansion. So the main defense in plants is to become mature, because then they have lignin, which make even more harder for herbivores to eat them. So these are the more vulnerable stages for a plant, and they have to defend with all resources they can. So there are many strategies. Some are chemical strategies, and the defense strategies I'm interested in is associating with ants, and that is via extrafloral nectaries. So another important thing that you probably listen later with Lisi Colli is that these stages, the young stages, is when most of the herbivory occur. So around 70% of total herbivory, one leaf alone, experiment during its lifetime will be during this stage. Okay, so again, with the nectaries, so you have these kinds of nectaries that I showed you in Ingas, but you have other types of other morphology for them. So this is a euphorbiaceae called Pausandra macropetala. It's one of the most common euphorbiaceae in here, in this area, and their nectaries are slightly different. So it will be here in the beginning of the petiole, and they will also have nectaries here in these tips of the leaves, and they are also active only during the stage of growth. So a lot of plants here have extrafloral nectaries. In fact, Don Finnern, professor at the U, he estimated that 30% of all plants in a forest have extrafloral nectaries. It's a lot, right? But I am guessing you guys never heard of facultative association. What you have heard, I would imagine, about plants and ants association is the obligated associations, which are so cool, but at the same time they're very rare. From what I've seen, it's something around 5 to 10% of all the plants in the forest have associations, obligated associations. So let me just illustrate it with another plant. So this is a Irtella mimercophyta, which is a Chrysobalanaceae plant, and these plants, this species has this dumaceous, which is like a round structure in the base of the leaf where the ants will house in. So I hope you guys can see. Let me show you. Tiny little ants. It's a genus called Alomerus. There are two kind of species that will house in this plant. So it's Alomerus orthoarticulata, and the other one, I just can't remember the species, but it's another Alomerus. So, and these domaceous are a very good way of getting defense against herbivores, because the plants are living in there, so they're always there for the defense. Whereas the nectaries is a little bit different than, because the ants will have to find a nectary that is active and then start protecting the plants. So that's the basis of my research. I'm interested in these, but I'm mostly interested in the evolutionary theories related to extrafloral On my PhD, what I'm trying to test is the hypothesis about the evolution of these extrafloral which in my hypothesis is associated with the membracids. So come here, let me show you what membracids are. So here is a young leaf, and I hope you can see these insects. There are two tiny insects in there. These are membracids. So they produce, in the tip of their abdomen, they produce nectar. It's not nectar, sorry. The tip of their abdomen, they produce honeydew. And this honeydew, it's equivalent to nectar in its ecological function, because it's also attract ants to plants. So here you see two adults, and this is a young one, a younger one, and ants haven't found them yet. So my hypothesis is that membracids and extrafloral nectaries are ecologically equivalent on attracting ants, but extrafloral nectaries are cheap and safer resource for plants to attract ants. My evolutionary hypothesis is that first plants have evolved extrafloral nectaries as structures, and then on the second hand, for the nectaries not to compete with the membracids, they have evolved defensive strategies against these membracids. So for that, I am first doing a census on all the plants that have, and the plants in here. So I have a group of plants that have extrafloral nectaries and a group of plants that do not have extrafloral nectaries. So I'm testing that on those plants. And then secondly, I have experiments. Let me show you the experiment. So one of my experiments is to try to measure how advantageous it is to have membracids versus having extrafloral nectaries. So for that, I choose a plant that is very common here, which is this plant here called Dugetia flagellaris. It's an Annonaceae plant, and it does not have extrafloral nectaries, but commonly we see membracids on this plant. So what I did was chose many plants that have membracids, many plants that do not have membracids, and I added extrafloral nectaries, artificial extrafloral nectaries. So as you can see here in this plant, this is my nectary treatment. So I put these tubes here. It's full of my artificial nectar, and then I'm letting here during leaf growth to see if ants will come. So as you can see, it's working. And and then I'm letting here, and after the leaves finish its growth, I'm going to compare how much these leaves were eaten versus the plants that have membracids. So another thing that I need to define, need to measure is, okay, maybe the ants are coming here, but are they actually efficient against predators? So another part of my experiment, in the same plants, I added an artificial caterpillar. So here, as you can see, I put it like a modeling clay caterpillar, which is very similar to the caterpillars we see around. And I waited 24 hours to see how much these will get eaten. So I don't know if you're going to be able to see. Can you see little bites here? So these were bites made by the ants that were coming on the nectaries. So it's working too. And let me tell you, most of the time the experiments do not work. And this is very fortunate, this one is working, so that's why I need to celebrate more. So a third treatment is not to have any of those. So membracids, so I have a group of plants, which is my control plants, that does not have membracids nor extrafloral nectaries. So let me go show you there. So this is my control group, and it does not have neither membracids or extrafloral nectaries. So I'm also measuring the caterpillar, the predation experiment, and I'm also measuring leaf consumption. And here is still in the very early stage of growth. So we'll have. This is what I've been doing in Brazil since April, and it's a lot of work. But I'm very happy to be home because this is my country, and I really enjoy being in the forest, although there are a lot of adventures that we go around here. But I'll be very happy to be back to Utah, and I'm guessing I'll be there in November. So I'm happy to show you the ants I've collected and whatever other things you would like to see that I have back in Utah. I'm very honored to be able to talk to you guys. Thank you, Yael, for this opportunity, and thank you for buying my crazy idea. And I hope you guys have enjoyed your trip. And I'll be happy to answer any of the questions through Skype. Bye.